Good morning, dear students. We are supposed to look into the next topic of our unit, that is the DNA damage and the repair mechanisms. Now, in the initial topic, what we had discussed, as you know, that how the DNA replication takes place. So, in a normal positions, in a normal cell cycle, the DNA on its own, as the cell divisions come to an end, continuously the cell cycles go on happening, and with the cell cycles continuously repeating, the DNA undergoes a process of replications to form a new set of DNA, or we can say a new set of chromosomes, which helps in the mitosis of the cell multiplication of the cell. Now, as we have taught or we have learned earlier that how the cell undergoes replication. But one thing one has to remember, that in natural conditions, the DNA might also face some problems, and that may cause the damages. So what are the reasons or what are the causes of problems we will be discussing in this topic, and how does the DNA overcomes on its own naturally the repair mechanisms during the process of the replication of DNA, what we people have to discuss in this topic. So what are the learning outcomes of this unit, what we have to discuss? So when we see this topic, after the end of the topic, the students will be able to explain how the errors during the replications are repaired naturally. So this is a natural mechanism where the DNA has its own capabilities to repair on its own. So the very first, what it is, the DNA should identify that something is going wrong. Something is wrong in the sequence of DNA. So what is wrong in the sequence of DNA is to be identified first. So who identifies? Again, it's the DNA who identifies its own problems. So when it identifies its own problems, whether the sequence is in a proper way or not, it identifies it, and accordingly the repair mechanisms are built up by its own. So how is this mechanism, and what are the different kinds of DNA damages we'll be discussing later. Then the students will be realizing that the most mistakes during replication are corrected by the DNA polymerase. So DNA has its own enzyme, what we call as DNA polymerase. So this is the enzyme which corrects the mistakes of the DNA and the replication by the post-replication repair mechanisms. So if at all during the process of replication, before or post-replication mechanisms, if at all the DNA recognizes that there is a mistake, there is a mismatch in the sequence of DNAs, the DNAs are repaired again. And if they remain the same, that will cause a mutation. That will cause another property or another characteristic property of the DNA. So, when replication mistakes are corrected, they may result in mutations which sometimes can have some serious consequences. So, you will realize or you will recognize that as the DNA replication or the repair mechanisms in the DNA comes to an end, a new sequence what results in the formation of a mutation. So in the nature, you know that there are various genetic disorders like PKU, phenylketonuria imbecility, we have SCA, sickle cell anemia, we have Creutzfeldt syndromes, many more. There are lots of diseases which are occurring in the nature, which they are just because of the mutations. So this is what the outcomes of the topic, the DNA damage and the repair mechanisms will be. So the next we will be discussing about what is the DNA damage. So where DNA in a living cell is subject to many chemical alterations. So as you students know that the DNA is a full set of chemicals. That chemicals, when they are subjected to function in a natural, in a normal conditions, they serve normally. But if there are certain alterations in in the sequences, in the chemicals, in the properties or in the structures of the chemicals. So that causes alteration which leads to the mutation. Now, the genetic information encoded in the DNA has some uncorrupted forms. So what are the uncorrupted forms we will be discussing later on. So any chemical changes must be corrected. So if at all during the formation of a replication, in the process, if the DNA recognizes that there is a change in the sequence of DNA, the base pairs, they are immediately, naturally, they are corrected. So when they are naturally corrected, finally a failure of the DNA repair, the failure to repair DNA produces a mutation, as I have discussed earlier. So now, where we have to see what are the chemicals or what are the agents which are responsible for the cause of the damage of a DNA. So where the very first I should say it is radiation. So radiation is one of the very problematic case where it can cause a mutation, it can cause a damage to the DNA. So where you see a highly reactive oxygen radical produces during the normal cellular respiration as well as during other biochemical pathways also may lead to the formation of a damage of the DNA. So what the more major focus is to given upon is the highly reactive oxygen species. So what is ROS, highly reactive oxygen species? So there are several chemical reactions in the cell which are going on. So with the chemical reactions, it may be a simple antigen-antibody reaction or an enzyme-substrate reaction. So where it produces some end products, some byproducts, so raw products. So these, if in the excess, so see, this is very most important. So ROS, that is the H plus ions, the high amount of H plus ions may cause a damage to the DNA. So that is the ROS. Where the second is the ionization radiation such as gamma rays and X-rays. So now and then, because of the development in the technology, we see for the X-rays and many things, we use the various gamma rays and X-rays. So if in a very prescribed limit, if the X-rays are exposed to the body, they don't cause any mutations. But if you are exposed for a multiple times and for a very high exposure of the X-rays or the gamma rays, they may cause a mutation, damage to the DNA. Apart from this, you can see various ultraviolet rays, especially the UV rays, ultraviolet rays with you can see 260 nm wavelength. They may also cause the damage to the DNA, and that are absorbed strongly by the DNA, but also the longer wavelengths, that is UVB, that penetrates the ozone shield also, and it may cause the damage of the DNA. So these are the first agent, that is the radiation. The second agent, what you can see, is the chemicals in the environment. So now and then, today we are exposed to various chemicals. They may in the form of gaseous phase, or sometimes even we know that sometimes we are used to the body sprays right now what we have. So even sometimes these body sprays also can cause or lead to damage of the DNA. So they are either the aromatic hydrocarbons, including some of the cigarette smokes also. So just merely smoking a cigarette and the smoke which is released may also cause a damage to the DNA. Apart from this, you can see various plants and microbial products. For example, alpha toxins. So these aflatoxins also are very important. If in a very slow amount they are available. If the amount of aflatoxins increases, they may also cause a damage to the DNA. Apart from this, you can see chemicals used in the chemotherapy, especially the chemotherapeutic agents which are used for the treatment of cancers. So they are also lethal. They also cause a damage to the DNA. So that damage to the DNA is of most importance. The next is the types of DNA. What types of damages to the DNA occur. So you can see the very first is single base alteration. So single base alteration, there are again different kinds. So it is depurination, the deamination of cytosol to the uracil. You can see the deamination of the adenine to hypoxanthine. You can see the acylation of base pairs, the insertion or deletion of the nucleotides, the F, that is the base analog incorporations also may be a part of the single base alterations, and this is a type of the damage to the DNA. The next is two base alterations. So it may be the UV light induced thymine, the pyrimidine dimer, what is also called as. It is a bifunctional acylation agent, cross linkages which crosses. So this is the base pair. You can see the two base pair alterations which leads to the damage of DNA. The third is the chain break DNA damage mechanism. So where during the ionization, the radiations, the entire chain of the DNA, it may be damaged. Next, the radioactive disintegration of the backbone element. So as you know that in the process of replication, one strand of the DNA forms the backbone. So that strand also may be damaged, and with the severe damage of the backbone, you can see it cannot synthesize, or it cannot again reproduce the next strand, another replica of the DNA, and the sequences may change. Next is the oxidative free radical formations. So what we call as ROS, oxidative free radicals. So these ROS also may form a damage to the DNA. The next is the cross linkages type of DNA damages. So between the base in the seams of opposite strands, and the second is between the DNA and the protein molecules, especially the histone molecules are one of the major reasons for the damage of DNA, and these are the four types of DNA damages. So the first what we are supposed to see is the DNA repair. In the first case, how the DNA repair mechanisms are going to take place. So you can see the DNA repair can be grouped into two major functional categories. The one is direct damage reversal. So during the entire process of replication, so if at all there is a damage to the DNA, directly during the process of replication, the damage can be reversal. Again, it can be reverted back. The same base pairs can be removed, and the new, fresh, original base pairs can be reformed there, and that is what we can see is a direct damage reversal. And next is the excision of the DNA damage. So that small piece of DNA damage is excised, means cut upon, and it is removed off, and it is replaced by the small piece of DNA. So these are the two kinds of repair mechanisms what we people are supposed to see. So you can see the very first, the direct damage. So where you can see a DNA, a very small piece of DNA, you can see here. This is a very small piece of DNA which is having a damage. So where it is synthesized, you can see a new pair of DNA damage is synthesized and it is replaced. So this is the base pair excision repair. The next what you can see is the direct repair. So during the formation of DNA, the DNA is directly repaired. And the third what you can see, the nucleotide excision repair. So you can see this is the repair mechanism, and where you can see the nucleotide excision, the base pair, it has been damaged and it has been repaired. So this is the nucleotide excision repair mechanism. So the direct repair damage reversals. So where you can see the direct reversal of DNA damage is by far the simplest repair mechanisms that involves the single polypeptide chain, where within the enzymatic properties which binds to damage and restore the DNA genomes to its normal state, it is a single reaction step. So the major polypeptide involved in these pathways are one, DNA photolyase. It is one of the major important polypeptide. So the enzyme responsible for removing the cyclobutane pyrimidine dimers from DNA and a light dependent process called as a photoreactor. So sometimes when I said that the DNA repair mechanisms which are conducted because of light, UV light, ultraviolet light. So it is the DNA photolyase which is responsible for the repair mechanisms or the direct reversal. Next is O6 methyl glutamine DNA methyl transferase 1 and 2. So MGMT, that is DNA methyl transferase, also called as DNA acyl transferase. So removes the modified base like O6 acyl glutamine and O4 acyl thymine, which is replaced or repaired. The photolyase protein is not found in the living cells. However, the DNA acyl transferase are widespread in nature. They are available freely in the nature, and immediately the acyl transferase can repair the DNA directly. So the next is direct DNA damage, where I explained. So you can see this is the normal DNA strand, and where you can see the dimer, the pyrimidine dimer, it is damaged because of the UV light, ultraviolet light. So you can see a common complex of DNA with a photoreacting enzyme. So photoacylase, just now as I said, that enzyme is very important. So that immediately the enzyme, it comes there and it starts see the absorption of light, it absorbs, and you can see the UV light, it is absorbed, and the release of enzyme to restore the DNA. So the now normally with the help of these enzymes, the DNA, it can be again restored, and the DNA repair mechanisms immediately. So what we call as a direct DNA repair mechanism takes place. So the next is the excision of a DNA damage. So there are again 4 kinds. So first is the base excision repair, also known as BER. The second is nucleotide excision repair, that is NER, nucleotide excision repairs. The third is mismatch repair. Mismatch. So suppose the sequence of DNAs are mismatched, and that kind of sequences are mismatched, it can be repaired. So this is also called as MMR, mismatch repair mechanisms. And the next is the strand break repairs. And sometimes what happens, even an entire strand, it may get damaged, and that entire strand also has to be repaired by the DNA. So that is the strand repair mechanisms. So where in these reactions nucleotide segments contains base damage, the double helix distortions or a misrepaired. So bases is replaced by the normal nucleotide sequences in a new DNA polymerase synthesis processes. All of these pathways have been characterized in both bacterial cells as well as the eukaryotic organisms. So in both the cases, prokaryotic organisms as well as in the case of eukaryotic organisms, the excision DNA damage is repaired also. So whatever it may be, the base excision BER, it may be NER, MMR, or even the strand repair mechanisms, all these things can be repaired in both the cases, that is the eukaryotic cell or a prokaryotic cell also. So the base excision repair. So the base excision repair BER is initiated by an enzyme called as DNA glycosylase, which catalyzes the hydrolysis of N-glycoside bonds, linking particularly types of chemicals altered base to the deoxyribose phosphate backbone. So where the DNA damage is excised as a free base, generating sites of base loss called as apyrimidic AP sites. So the AP sites are substrates for AP endonucleases. So these enzymes produce incisions in duplex DNAs as a result of hydrolysis of the phosphodiester bonds, immediately the 5' or the 3' to each AP sites. So the ribose phosphate backbone is then removed from the DNA through the action of specific exonuclease, also called as deoxyribophosphodiesterase or DRPase. So finally, the DNA polymerases and a ligase catalyze the incorporation of the specific deoxyribonucleotides into the repaired site, enabling the direct base pairing. So immediately the base pairing is taken off, and the repair mechanisms in the case of base pair repair excision or the base excision repair mechanisms takes place. So this is how the base excision repair takes place, where you can see the C because of heating it is replaced by uracil, that is cytosine is replaced by uracil. So uracil DNA glycosylase acts here. You can see the repair mechanism has started. The excision, the cut is made into the DNA, and the DNA polymerase enzyme and the DNA ligase again repair back, that is replaces the uracil molecule into the cytosine molecule. So the base excision repair of DNA. So the enzyme uracil DNA glycosylase removes the uracil created by the spontaneous deamination by cytosine of the DNA. The endonuclease cuts the backbone near the defect. So you can see the endonuclease have cut the single strand of DNA where the damage had taken place, and the defect is filled by an action of DNA polymerase. So that the DNA polymerase again comes into action, and the mechanism of replacing uracil to cytosine takes place, and the strand is rejoined by again the enzyme DNA ligase. So this is the base excision repair mechanism. The next is nucleotide excision repair, NER. So the mechanism is used to replace the region damaged by the DNA up to 30 base pairs in length. So what we spoke initially, the BER, it was a single base pair. So up to 30 base pairs, a very long chain of DNA with the help of the nucleotide excision repair, NER, also can be changed. That is also can be repaired. So where you can see the common causes of DNA damages include the ultraviolet light, which includes the formation of cyclobutane, pyrimidine dimers, and smoke, which causes the benzo, alpha, pyrene, guanine abducts. So these are the damages what are called the damage causing agents, the cyclopyrimidine, or you can see the smoke in the form of benzo, pyrene, guanine abducts. So where apart from this, the ionizing radiation, cancer pyrimidic agents, and a variety of chemical forms in the environment causes the base modification, strands break, cross linkages between bases on opposite strands or between the DNA and protein of numerous other defects. So these are repaired by the process called as the nucleotide excision repair, NER. So the NER is much more a complex biochemical process rather than the BR, base excision repairs, especially in the eukaryotic cells. Several genes are required in a multiple steps process during which the order of assembly of the DNA proteins provide an enzyme complex that discriminates the damage from the undamaged part of the DNA. The nucleotide excision repair, NER, how does it work? So where you can see, this is the normal set of DNA where you can see a 3 prime, 5 prime. So where you can see maximum 4 or 5 base pairs damage to the DNA has took place. So where it is recognized first. The recognition is of most importance, and after the DNA is recognized where it has been damaged, so where you can see the unwinding. So at that place, the strand of the DNA has to be unwind, and when the DNA unwinds, there you can see in this location the oligonucleotide excisions by cutting the two sides on both the ends where the damaged part of the base pair, the nucleotide base pair has took place, has to be cut open, and after all you can see the degradation of the mutation takes place and the resynthesis, the damaged DNA is removed first of all, and when it is a process what we call as the degradation of DNA damage, and then it is replaced by the resynthesis and the relegation of the DNA. So how does it take place in eukaryotes, especially the cells and enzymes cut the third to fifth phosphodiester bonds, the 3 prime form the lesions, and on the 5 sides the cut is somewhere between the 21st and the 25th bonds. Thus a fragment of DNA in the 27-29 nucleotide long is excised. After the strand is removed, it is replaced again by exact base pairing through the action of yet another polymerase, and the ends are joined to the existing strands of the DNA ligases. So this is the nucleotide excision repair, NER, where you can see the damaged nucleotide is identified, it is replaced by, so these are in the E. coli, Escherichia coli, they are the 3 specific proteins, especially the UVRA, UVRB, UVRC, involved in the lesion recognitions and endonuclease resignations. So these are the enzymes which recognize first the base pairs where they are damaged, and when they are damaged, recognized, you can see they are cut open and the DNA polymerase 1 and the DNA ligase enzyme, they act upon and they again synthesize the new nucleotide base pairs. So this is NER, nucleotide excision repair. The nucleotide excision repairs produce most rapidly in the cells. Those genes are being actively transcribed on the DNA strands that is serving as the template for transactions. If RNA polymerase 2 tracking along the templates, that is the antisense mechanism strands, encounters the damaged base, it can recruit other proteins to make a quick fix before it moves on to the complete transcription of the genes. So this is the mismatch repair, MMR. So where you can see the mismatch repair corrected errors are made. The DNA is copied first. The DNA is copied for mismatch repairs. For example, a C could be inserted opposite to A, that is cytosine can be opposite to adenine, or the polymerase could slip or shutter to the insert 2 to 5 extra unpaired bases. So the specific proteins can be scanned with the newly synthesized DNA using adenine methylation with the GATC sequence as the point of reference. So the template strand is methylated and the newly synthesized strand is not. So the difference allows the DNA repair enzyme to identify the strand that contains the errant nucleotide which requires the replacement of the DNA strand. So if a mismatch or a small loop is found, a GATC endonuclease cuts the strand bearing the mutation at the site corresponding to the GATC. So an exonuclease then digests the form the GATC through the mutation, thus removing the faulty DNA. This can occur from either end if the defect is bracketed by the 2 GATC sites. This defect is filled in by the normal cellular enzymes according to the base pair rules. So the mismatch repair, you can see what I had explained right now. So this is the strand where you can see a single strand cut by a GATC endonuclease. So this strand is cut, the defect is removed. So this defect, you can see this, after this cut and a cut, you can see the defect of the base pair, it is removed, the defected pair is removed and it is repaired by a polymerase chain reaction. Again a DNA polymerase enzyme comes to action here and it repairs the DNA, and the recycling gated by the ligase. Again it is joined, the joining of the DNA base pairs takes place with the help of ligase enzyme. So ligase is a very important enzyme. So this mechanism corrects a single mismatch base pairs, that is C to A rather than a T to A, or a short region of unpaired DNAs. The defective region is recognized by the endonuclease. As I said, that endonuclease is the enzyme which identifies the defect and it makes a single strand cut and the adjacent methylated GATC sequences. So the DNA strand is removed through the mutation replaced and they are again relegated. So the mismatch repair mechanisms, MMR in E. coli, you can see. So this is where G and T, it is mismatched, because usually G makes its match with the C, GEC complex, we know. So where it has made a mismatch with the T, so you can see this is a break with the MUTH, that is a mutation H enzyme. So in E. coli, the protein MUTS, MUTL, MUTTH are required to recognize the mutation of the NICs. So they identify the mutation with these enzymes MUTS, MUTL, MUTH, and when it identifies, it makes a cut there. So other cellular enzymes including ligases, polymerases and SSBs removes the replace and replaces the strand. So the cut is made there again. After the cut is made, the DNA strand is removed and again it is synthesized with the DNA polymerase enzyme, and you can see it is again replaced back the DNA polymerase enzyme. So you can see here the newly synthesized DNA strand is replaces. So this is the mismatch repair, MMR, in the Escherichia coli. So the repairing strands break with the help of ionization, radiation. Certain chemicals can be produced, both single-stranded, that is SSBs, and a double-stranded break, DSBs. Single-strand break, SSBs, DSBs, double-strand breaks in the DNA backbone. So what is a single-strand break? A single-strand break, that is a break in a single strand of DNA. So as you know, the DNA is a double-stranded, double helical structure. So a single strand is broke open, and in the DNA molecules are repaired using the same enzyme systems, just like the various enzymes what we used initially, and that are used in the base excision repair, BER. The double-stranded break repair, so where you can see the DNA has to be cut open at various two different positions and the entire double-stranded DNA piece has to be replaced. So where there are two mechanisms by which the cell attempts to repair the complete break in the DNA molecules. So the very first is the direct joining of the broken ends. So this requires the protein that recognizes and binds to exposed ends and bring them together for ligating. This is the type joining, also called as non-homologous end joining, NHEA, non-homologous end joining. A protein called as Ku is essential for non-homologous end joining. The double strand repair mechanism. So there are two mechanisms by which the cell attempts to repair a complete break in the DNA molecule. The one is direct joining. For the broken ends, this requires proteins that recognize and binds the two exposed strands and bring them together for ligating, for joining back again. So this type of joining is called as non-homologous end joining, NHEJ. A protein called as Ku is essential for the NHEA, as I explained in the figure earlier. So errors in direct joining may cause various translocation of the sequences in the genome that are associated with the cancer. For example, the examples like Burkitt's lymphoma, a cancer, a Philadelphia chromosome in a chronic myelogenous leukemia, CML. It is a disease where blood cancer is responsible, and where you can see a beta cell leukemia, B cell leukemia. The other is the homologous recombinations. Here, the broken ends are repaired using the informat of the intact. Sister chromatids or the homologous chromosomes are also identified. Same chromosomes, if they are duplicate copies of the genes of the chromosomes oriented in the opposite directions, head to head or back to back positions. Two of the proteins used in homologous recombinations, they are encoded by the gene BRCA1 and BRCA2. So inherited mutations in these genes predispose women to breast or the ovarian cancers also. So this is the repairing strand for breaks. So where can you see a double-stranded break with the help of an ionization radiation or an oxidative damage, a spontaneous event. So here you can see the DNA at both strands it has been damaged. So where a strand breaking joining has to be take place and the homologous DNA has to be produced. So this is the meiosis, also involving DSBs, the double breaks where we have identified. So recombination between homologous chromosomes in meiosis 1 is also involved the formation of DSBs and their repairs. So meiosis 1 with the alignment of homologous sequences provides a mechanism for the damaged DNA. Thank you.